You've heard now really interesting stories at the cellular level about brain cells and how they function, and at the network level and how the brain functions. And I think now Dr. Derek Bowie will be talking at a much finer level of resolution, at the level of single molecules and synapses, I believe. And so I've introduced you a bit earlier. This is Dr. Bowie from Pharmacology and Therapeutics, and also a member of the IPN. And so, Derek, I'll hand over the platform to you.
Okay, thanks, Ed. Can everybody hear me? Yep, you can hear me fine. I think I'm fortunate in that someone's going to be running the slides for me. I think Meeting's going to be doing that. So I'm going to direct you as we go along. So she's also kindly translated the title of my slides, you'll notice. Just in case you think I'm skilled in Mandarin, it's not true. I had help doing this. Okay, so thanks again to everyone for the invitation. It was great to hear Boris's talk, actually. A really nice presentation, and nicely complements what I'm going to tell you about in the next few minutes. So I'm interested in the molecular underpinnings of brain disease. So I kind of believe that, you know, diseases occur at the level of molecules, and myself and Dr. Watt, who spoke a little bit earlier, both of us are actually neighbors in the same building. And myself and Alana are very excited, and Ed as well, Dr. Ruth Azer, very excited about the molecular defects are involved in brain disease and how also brain function. So what I'm going to tell you about is a kind of overview of that, which complements very much the very nice high-level work that Dr. Bernhard just told you about. So maybe you can move to the next slide. So this slide is just really to give you a kind of overview of what my lab does, and also a lot of people who are my colleagues at in the neuroscience community at McGill kind of view themselves. I have one little bit of exception I'm going to tell you in a minute, but my core interests are in the brain. So on the left-hand side, you see a sort of snapshot cross-section of the brain, and we're interested in just how the brain functions from its normal physiology. And we're also interested in disease. And one disease that we've got really interested in that actually nicely meshes in with Dr. Bernhard's work is we've got interested in neurodevelopmental disorders, but way down at the molecular level, which I'm going to just touch on very briefly at the end. In addition to trying to understand the normal physiology of the brain and how the brain is defective in different kinds of disease, such as epilepsy and autism, schizophrenia, etc., we're also interested in how genes impact brain function. It's becoming increasingly more and more aware to all researchers working in neuroscience that genes have a profound impact on brain function. And so we're particularly interested in trying to look at that in the next coming years. And in fact, we've got quite a lot of work developing looking at that. And I'm going to pick up on that at the end. And I know the audience isn't necessarily very familiar with genes and how they're regulated, but I think the way to think about them is there are packets of DNA which are inside all your cells and in your brain cells, which make important signaling proteins that determine how your brain functions. And those proteins, how they function, may vary depending on your genetic makeup. Okay? And so on the right-hand side, you'll see I've got this protein family called ion channels, which most of the audience won't be familiar with. But the best way to really think about ion channels is that they're the fundamental signaling protein in the brain. They're little machines, if you like, that transport ions, so charged atoms, across the membrane of cells. And when they do that, they actually provide very important electrical signals, and it's those electrical signals that I'm really interested in looking at. So just as a kind of general overview of things and how one could study brain function, maybe move to the next slide. So you've just heard a very eloquent talk about all the different kinds of techniques that you can use to image the brain. So if you're interested in how the brain, the morphological organization of the brain and how it's interconnected, then that's exactly the kind of techniques you would need to do. So all the work that's been done by Boris and his colleagues in the Imaging Center, McGill, are using all these kind of techniques. And what they're really good at is being able to looking at patients, identify structural defects and maybe even some functional defects depending on these techniques. My interest is in really time scale, which is on the millisecond time scale. And if you really want to understand the millitime scale, then what you really have to appreciate is that the brain is really an electrical organ. So it's really just like your muscle in your heart, it's electrical, and that's how it works. And one way to study that at the same level of the kind of techniques, which is shown here, is shown on the next slide. Here we go. And it's using a technique called EEG, so electroencephalograms. And the way this is done, and this is done routinely in the Neurological Institute, especially to diagnose patients for epilepsy, etc., is they put a sort of skull cap on. There's a bunch of electrodes you can see on the right-hand side with this particular patient here or volunteer. There's all these different kind of electrodes. And what you can do is if you have a reference electrode, and with respect to that reference electrode, you can actually measure tiny little microvolts of signals that are occurring in the electrical activity at the top of the brain. And from that, what we've learned over the many years is that the brain undergoes very dramatic changes in its electrical activity under normal conditions, when we're having a conversation, when we're visualizing light, when we're sleeping, and of course also when we're consuming different kinds of drugs, so alcohol, and taking medication, it can change these functions. And that's shown rather nicely on the next slide in this little movie that I managed to pinch from a colleague of mine. So if you just click the movie, so on the left-hand side, what you can see is a normal-ish EEG, which is going on. And then on the right-hand side, it's not a very serious scientific study, but it was taken from someone who was consuming some alcohol and also smoking some marijuana. And what you can see is, surprisingly actually in this person, there's a lot of excitation, a lot of excited activity going on in the brain. And so a long, long time ago, when I get into research and I was starting to look at these things and getting excited about it, what I wanted to do is really understand what's the individual, contributors to this kind of activity, because this is the whole brain working together. But what are the individual what's the individual makeup of these? And so to do that, we're going to move to the next slide. You actually have to study the brain cell by cell. You just move to the next slide. There you go. And to study the brain cell by cell, you have to use this kind of equipment. Here it's called electrophysiology, and perhaps Dr. Watt talked about this when she was talking about her work. This is Amanda Perosa, who's a graduate student in my lab, and she's using a technique called patch clamp electrophysiology. And if you show the next slide, you can see what Amanda can see under a microscope. So here's a typical kind of image that Amanda and people in the lab would see under a microscope. And what you can see is a nice big fat pyramidal type cell. It's got this kind of pyramid type structure to it, probably the cortex. And then you can see this out of focus patch pipette. So it's a glass pipette that comes in and can touch the cell. Yeah, if you just go back, yeah, you just back to one. It can touch the cell because that patch pipette is set up to very, very sensitive amplifiers, which amplify the electrical signal. Then you can actually measure electrical activities for these individual cells. And what we've learned over the years that I've been doing this kind of technique, if you now move to the next slide, if you don't mind, is that all cells, just like every individual person in a room, we all have our own personalities, right? And so do neurons. So individual nerve cells, they all have their personality. And here's 4 personalities for you that we recorded from some of the cells, which are in very important brain regions in the brain. So on the left-hand side, you'll see 2 cells from a region of the hippocampus. So the hippocampus is involved in learning and memory in the brain. And you can see that one cell gets excited, the one on the right-hand side gets excited and gets excited for long periods of time. Well, there's the other one who's got a more of a graded excitation, these kind of electrical responses you get. And that's from a region of the brain. I don't know if you can see it, maybe you can point to it. It's little green kind of slither at the top of that cross-section cartoon. You just see up there, it says hippocampus. Maybe you can point to it. Anyway, it's in the hippocampus, this little part of the brain that's very, very important for learning and memory. On the right-hand side, you can see you get 2 other cell types, which are found in that region of the brain, which is to the back of the brain, called the cerebellum. And I think Dr. Watt probably told you an awful lot about the very beautiful morphological work and electrophysiological work she does there. And you can see there's 2 different cell types there, down at the bottom of the slide. And you can see that they have quite different properties. So they have different personalities. And that's something that I'm really excited about, because that tells you something about the molecular makeup of these cells. If you go to the next slide, it's not just in the electrical properties of these cells, but also the chemical signaling that these cells undergo. And so what I've done now is taken the same cells at the top part of the slide, is the same data that I showed you before, but underneath now you can see in red and black, so all these experimental traces which are showing that go along the bottom of the slide are responses due to synaptic transmission. I appreciate that many people in the audience may not fully appreciate what that means, but it's this chemical transmission that occurs between individual neurons. And what you can see is that the fidelity of that chemical transmission is different for different cells. So not only is their electrical signaling different, but also their chemical transmission is different. And so all of these together give you that very complex electrical activity you see when you measure with electrodes placed on the skull of individual neurons. So we're interested in trying to understand what causes all of that diversity in these different types of neurons and how that signaling may go awry in different kinds of diseases. So maybe go to the next slide. So what I'm going to tell you about is just three of what I think are the most important signaling proteins in the brain. And it turns out we just happen to work on these three signaling proteins. Incredible coincidence, as you can imagine. The one on the left-hand side is called a sodium channel, and that gives rise to that very rapid electrical activity you see when these different types of neurons fire. And then the two bigger proteins, which are shown on the right-hand side, the one that's called inhibitory GABA, then that's like the traffic lights that shut down the movement of activity in the brain, whereas the one on the right-hand side, which is called the glutamate receptor, then that drives all the excitation in the brain. And so all of your brain activity is a kind of balance between these three different families of ion channels and how they work together. And so you can imagine in any disease scenario that there's bound to be some defect in one or all three of these different signaling proteins because they're so predominant in the brain. And that's why we study them all, okay? Now we've got structures of these, so we can actually have a prediction of how we think these proteins might move in real time. And if we maybe move to the next slide. And so here's a little cartoon of how we think the glutamate receptor, so this is the excitatory receptor, how it works. And what you'll see is those little two things are the neurotransmitters, these chemicals that bind to it. And this whole thing is happening in the orders of nanoseconds. You can see that the protein is crunching down on a rapid time scale, and then that's leading to an activated state of the protein, and then it inactivates, undergoes another change, and then it activates. And that's something which is happening in your brain on an incredibly rapid time scale all the time, right? And in individuals, what we're learning in disease is that that process is defective in a lot of kids, especially in epilepsy and also in autistic disorders. Another property of these ion channels, which is also shown on the next slide, is that they've got a central ion channel pore. So they've got a hole in them like a barrel that has a hole in it, and they let these charged ions to go through it. And you can simulate what that looks like. And I'm going to ask Mei Ting to actually just click on the thing, the movie. And so what you'll see, this is a molecular dynamics simulation of what's going on. You can't ever see these ions going. It's so fast, right? But if you put a big fat ion in the middle, so there's a spermine, you see this long charged molecule, which is stuck in the middle of this pore, then what you can do is you can slow the movement of ions through the pore, and you can actually visualize what actually happens. So if you hang on a few seconds, what you'll see that, remember this is happening over about 25 nanoseconds, you can see that eventually, bang, they get through, and they're out through the pore. And that's how our brain signals. That's exactly how it works. So it's working on this very, very fast femtosiemen, nanosiemen time scale. And the diseases tie into defects at this time scale. So I'm just going to go to my last slide. So why is all of this biophysical information interesting to us? So because on the left-hand side, you can see we now have full-length structures of these proteins, which are fundamental to how brains work, so how we learn, how we see, how we hear, and you know, how we process information at very high levels. Then what we've been learning in the last 5 or 10 years is that individuals, so on the right-hand side, you can see that these are the individual subunits that make up this 4-subunit protein called the glutamate receptor, which is very dominantly expressed in the brain. You can see that we've mapped onto that structure different mutations which are found in children with autism. And what you see is that based on the color, I don't know if you can see the colors, and then the positioning of where these mutations are, that most of the mutations are actually low down in the protein structure, right in the plasma membrane, so right on the surface of the cell. And that's where most of the defects that seem to be found in autistic disorders. So the autism disease isn't just one genetic defect, but it's all of these defects together. But without knowing the structure of the protein, then there's no way to really understand how all of these come together in this kind of map in trying to understand how this functions. So now we have these mutations, we can make these proteins and then study the function and try to understand how it works. So here's just the last thing I want to leave you with. So now that we've done this work, we've learned that these mutations can cause a gain of function in the protein or a loss of function in the protein, but yet lead to the same disease. And that's kind of perplexing, right? But what it means is that because we have drugs which will boost the protein function or reduce the protein function, then what's going on in the clinics now is that we are sequencing patients with autism, identifying the mutation. And if the mutation is a LOF, that's a loss of function, then you give it a drug for a gain of function. But if the mutation is a gain of function, then you give a drug which is a loss of function. So that means that when we're thinking about treating autism, then we may be actually giving very specific types of drugs to different persons based on the profile that we see all the way down at these molecular details. So I think that's something very exciting, and especially in the context of McGill, where we have people like Boris, who's doing this very elegant work all the way up at the level of the brain, down to people like myself and Alana and Ed Rutaser and myself, trying to work out the nitty-gritty down at the molecular details. Then I think we're working towards each other to try and solve the molecular basis and then fundamentally the system nature of these diseases. So that's really it. I'm just going to go to my last slide. So if anybody's interested in this work, so we do quite a bit of mathematical modeling. We do electrophysiological recordings. So the students tend to come from mainly physiology, biology background, but we have students from maths. We also have students from chemistry. And we've also got a student coming through right now who is bioengineering background. And so we kind of draw interest from a different kind of group of people coming in. So if you're interested in learning more about our lab, here's our sort of snapshot of our lab website. And here is the web link if anybody is interested in going to learn a little bit more about it. We've got a bunch of pages where you can browse and see what we've published and the kind of people that are in your lab. So that's really it. So thanks for your attention, and I'll be happy to take any questions.